Greetings and welcome back. So, we're in the process of trying to explore the facts of evolution and the ingredients of evolution. And so, in the previous vignette, what we talked about was the fact of variation. And now we're going to turn to the fact of heritability. That is to say, that this ambient variation, which is present in a reproducing population, can be inherited by the offspring. Okay, and so the point of this brief vignette is just to walk through that. So I will go at a relatively rapid pace. The outline is that I want to start out by giving you some background and really having to do with Mendel and genetics. It's a story that probably many of you know in a superficial way, in a good way, but it's much deeper and I think more interesting than the sort of high school version. And we'll then talk about the reconciliation between Mendel's abstract particles of inheritance, which we now call genes, and the modern molecular view in the form of DNA. So, so the rise of genes and then DNA. I think, one of the things that I believe that maybe sometimes we forget or don't remind ourselves about is how abstract the discovery of Mendel was. So, as you can see here, and it's a well-told tale. Mendel was a monk in what is now Austria, or maybe the Czech Republic. I can't remember anymore. And the beautiful story, I think, is told best by Eric Lander. So if you go to MIT's sort of freshman biology course, it's taught by Lander and Weinberg. I think it's like lectures 5, 6, and 7. There's a priceless 10-minute vignette by Lander in which he talks about Mendel and what you might call the synthetic biology of the time, where in the Austro-Hungarian Empire, they were trying to compete with the Spaniards in terms of quality of wool and things like that. And the concept was, how do we do better on inheritance, like in a certain sense, in constructing animals and plants more to our liking. And so he, meaning Lander, argues that this was a biotechnological incubator, that they were intentional in trying to grab young students and get them trained in physics and math and chemistry and biology, such that they would be able to do for the study of inheritance. They used such grandiose language as what Newton had done for mechanics. And I think it's all the more ironic that that's exactly what played out. So, I think I'll pass on this, but I highly recommend that you watch this little segment from Lander. So what was going on? Well, Mendel basically spent some number of years getting his experimental system in place. It wasn't always peas. He tried other plants. And what he was interested in was basically trying to find an organism that would breed true. And when I say breed true, I mean that when he performed crosses of what we would now call homozygotes, they would stay homozygous, and then he could cross them. And I think he looked at 7 or so distinct characters. Interestingly, if I remember correctly, the number of chromosomes is 7. So, he basically found traits that were on different chromosomes. Again, I'm a little bit off in terms of my memory. I haven't read a biography of Mendel in a while, and I haven't reviewed these things. So, what was the upshot of what he was doing? Well, he was comparing sort of what you might call macroscopic traits that were ostensibly very different. So you can see, the flower color is a very obvious one. Another one was the form of the seeds, whether they are round or wrinkled, and so on. And so what he did is once he had this model system, then he would take the variants that bred true, and then he would cross them, as you see in producing the F1 generation. And then he crossed those, leading to the F2. And I think that that's where the surprise and the richness of the phenomenon really revealed itself. And I think, there's a great merit, I find. I mean, I've always found, since I was 17 years old, really. Niels Henrik Abel, one of the great mathematicians ever. We're living in a pandemic. I think it's good to remember that mortality once upon a time was terrible. So Sadi Carnot, as in the Carnot cycle, died at age 36 of cholera. In the case of Abel, I think he died at age 27 of tuberculosis, if I remember correctly. So, at any rate, Abel made this remark: read the masters, not their pupils. And I think that in mathematics, there's super merit to that. You know, you can still, with advantage today, go and read Mendel, or you can read Gauss's disquisitions on curved surfaces, or read Riemann's paper on generalization of what Gauss had done to manifolds. And all of those things are incredibly interesting and thought-provoking. I also think that the paper from Mendel is really quite beautiful and quite interesting. And, you know, I love the way that Lander talks about this, because what he points out is that the data was not what you all learned once upon a time. You know, look at the right-hand column here. So basically what we're looking at is the ratios that he discovered, and none of them are 3-to-1. You know, they're approximately 3-to-1. And what Lander has to say is that it was part of the genius of Mendel to understand that the data was trying to tell him something deeper, in the sense that 3-to-1 had some sort of bigger significance. So, you know, I just point out, I grabbed this page from the paper. You know, the form of the seed from 253 hybrids, 7,324 seeds were obtained in the second trial year. Among them were 5474 round or roundish ones and 1850 angular wrinkled ones. And let's see if that, there you go, seed shape. So this column, or sorry, this row. And, you know, the ratio is 2.96 to 1. So, you know, we all know this, and that's the problem is, you know, we learn these things, and there's like a mythology when we learn them. It's like a formula almost, where there's Mendel, and he did crosses, and then he measured 3-to-1, and there's a Punnett square, blah, blah. But that's not science. Science is the confusion of the moment, and Mendel trying to reason out, you know, what could be behind this weird thing where in the F2 generation you get an approximately 3-to-1 ratio. And, you know, would I have thought of the notion of recessive traits and that sort of thing? I don't think so. I don't know. I mean, there's times in mathematics and physics where I can imagine myself having an idea, but somehow, I don't know, this one seems very distant to me, that Mendel was able to figure this out. And as I say here, this is really my favorite kind of science, because it's indirect, and it's predictive, and it's inventive. And the upshot is, you know, he had an abstraction. We now call them genes, but, you know, he's saying that there's genes for various traits, and there's 2 copies of them, and they mix in some way with this rule, which we call the Punnett square. So over in cell biology, there's a concomitant development, and this is illustrated by these beautiful drawings from Walter Fleming. So people like Fleming and Boveri and Sutton were studying cell division, and they noticed that there were these structures. You know, in physics, we like the notion of ons, O-N-S, electrons, phonons, plasmons, magnons, spinons, blah, 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 blah. And in biology, we like our somes, which, you know, means bodies. So what is this thing? Well, it's a chromosome. What does that mean? It's silly, really, if you stop and think about it. It means colored body. So, you know, there were these things that you could stain, one could watch them, and during cell division, one noticed that they segregate in pairs. And so, you know, people like Boveri were, in a way, emboldened to make a hypothesis, which is maybe these chromosome things are the carriers of those abstract particles of inheritance that Mendel had been talking about, which is, you know, beautiful, very interesting supposition that was, you know, borne out, and skip this, but was really borne out in the studies of Thomas Hunt Morgan and his team of researchers. So I mentioned this in the previous vignette on variation, and the idea is that certain mutants were found, and the clever thing that they did in the Morgan laboratory was to cross flies that had different mutations or different phenotypes. So let me see if I can be more concrete about that. So, you know, you could cross flies that have different wings and different eyes and so on. And the thing that they were exploring was was the probability that two different traits would go along for the ride together. In other words, the offspring would carry both of these traits. And it's a little bit too sophisticated, and I don't mean that in the sense for your minds, but I mean for me in a short vignette to go into in detail. But here's the concept, which is that there's this process of crossing over, which I'm sure all of you have heard about. So you can see we have the red and the blue, and then they get mixed up, where there's a crossover in the sense that part of the blue joins up with part of the red, and now we're prepared. Let's see if I made a slide for this. Yeah, we're prepared for the inside of Sturtevant. So what he argued is shown on the right-hand side, which is if I have these two different traits that are nearby on the chromosome, then they, during a crossover event, they have a higher probability of going along for the ride together. That is to say, to be inherited together. If they're farther apart, then when the crossing over event occurs, it's more likely that the cut will take place between them, and so they'll segregate independently. And from this, Sturtevant was able to basically set up a chromosomal map, which has stood the test of time. I mean, I think that this is amazing because, and again, goes back to my favorite kind of science. Even though we live in this era where we're very, very excited about direct evidence, I still find a certain beauty to when we're able to work out things by indirect reasoning. And so here, by basically making this map on the basis of frequencies, it's a probability argument. One was able to generate a map of the Drosophila genome, which modern sequencing shows us to have stood the test of time. Okay, so let's move forward another 20 years after this. So what have I said thus far? I guess I've said that in some ways the Morgan experiments really gave a lot of credence to this idea that the abstract particles of inheritance of Mendel and these colored bodies, the chromosomes from cell biology, were the same story. Now, what's the molecule that carries that information? Of course, every high school kid can now recite that, and the answer is DNA. But how did we come to that knowledge? Knowledge is hard. How did we actually come to the point where we believe unequivocally that DNA is the carrier? So one of the sets of experiments was done by Griffiths. Really interesting. Again, I don't really have that much time to go into it, but the idea is that there were two different strains of a bacterium, that one of which is virulent and one of which is not. And if you take the virulent strain and put it in a mouse, it will kill it. If you take the non-virulent strain and put it in a mouse, it will not kill it. What they did is they then killed the bacteria that were virulent and injected that into a mouse. The mouse lives, so that's the middle column. But then the interesting one is on the right, which is to say that if you take the dead virulent strain bacteria and mix it with the non-virulent, now the mouse will die. And this is basically because something from the boiled bacteria, if you like, was brought on board into the bacteria that were previously not virulent and rendered them virulent. And Oswald Avery and his co-workers did a really interesting thing. What they did is shown on the right-hand side, and I really love this. So what they did is they used a protease, they used an RNase, and a DNase, and each one of those will destroy one of the molecules, and then they ask the question, is the virulence still there after this protease treatment? And the answer is no. After RNase treatment, no. But after DNase treatment, then the transforming principle is no longer there. So the conclusion was that DNA was the genetic material, and this is a graph from the original Oswald Avery paper, and the chemical analysis is basically telling us about the composition, and the composition is consistent with the deoxyribonucleic acid. I think that's the thing I want you to take away. So this was a very fast and probably inappropriate attempt to just acquaint you with the hard-won and beautiful knowledge in going from Mendel's peas to the structure of DNA. Let me just say one more word about sort of what sealed the deal on the subject of his DNA, the genetic material, was experiments on bacterial viruses. So I'm showing you the bacterial virus life cycle. The phage shown in the upper right infects a bacterium. What happens after that is that the DNA is now inside of the host bacterium. The bacterium knows what to do with that genetic information. It copies the DNA. It also synthesizes the proteins that are encoded in that viral genome. The capsids assemble. I can't even begin to say how amazing that is. I'm just saying words, capsid assembly, but stop for a moment and think about that. 100 little perfect viruses synthesized by self-assembly. The DNA is packed through an ATP-consuming motor, and then the cell lysis and these go on and repeat their deed elsewhere. So Alfred Hershey and Martha Chase did this experiment known as the Waring blender experiment, in which they did a brilliant thing. They used radioactive sulfur to label the proteins. They used radioactive phosphorus to label the nucleic acid. They allowed the phages to infect the bacteria. Then they used a blender to remove the phages from the cell. And then they did an assay to ask, where is the radioactivity? And what they found in the latter case was that radioactivity was in the pellet. In other words, they centrifuged the pellet down, which is the cells. In the top case, they did not find radioactivity because that was present with the sulfur, which is the viral capsids, and is not present inside of the cells. On the other hand, after infection by the phages, now the bacterium has radioactive phosphorus. So this was really, the smoking gun, if you like. So shortly thereafter, 1953, Watson and Crick, it's all well known, I'm not going to belabor it, but they worked out the structure of DNA. And, there's this comment at the end, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. I think I'm not going to tell you about the Mendelssohn-Stahl experiment, but it's an amazing thing. You can take a look at these two figures. You could just read this, but go to the thing that I cite at the bottom. It's a very nice description of the Mendelssohn-Stahl experiment, actually with a real connection to Caltech and the Marine Biological Laboratory. People might find that interesting to learn a little bit more about. They were actually, if I remember correctly, at the Catalina apartments, and Mendelssohn and Stahl pulled off this, amazing, brilliant, clever, in modern terms, kind of low-tech experiment, but, with deep, deep insight to come out of it. I'm also going to skip this about Larry Delbruck. So, last point. So, I basically here am talking about heritability. So the previous vignette was about variation. This one is about heritability. And what I want to note is that we already talked about the variation in the beaks of the finches, for example, on Daphne. And I told you about the methodologies used by the Grants in order to measure beak sizes. And this was the kind of thing that came out of their analysis, which is to say that there's variation. And now, this is the denouement, the key insight, which is now if you ask the question, what's the relationship between the beak depth of the parent, which is the x-axis, and the beak depth of the offspring, there's this nice correlation, which I'm going to offer here as a approximate qualitative thing that you can go look into more deeply about the heritability of this particular trait, which, in terms of the facts of evolution, which is really our topic, the fact of evolution is that first there's variation, which is the clay from which evolution is sculpted, and that variation is heritable. So from one generation to the next, the traits, my kids clearly have some traits that they acquired because of the heritability of variation from me, for example. So summary. What have I tried to tell you? I've tried to, above all, it's a qualitative statement. I wanted to make it clear that there's this notion of variation, which is a fact of evolution. Next, heritable variation, fact of evolution. It's also, this vignette was meant to give just a little bit of a feel for the tortuous path to understanding, and also the importance of abstraction. We now call what Mendel discovered genes, but, it's not at all true to say that which passed in the, 1860s was really at all what we think of as the modern conception of genes. We have another 150 years of work that has put meat on that. So the next vignette will be the final one about this notion of, what are the ingredients of evolution, and that'll be on the forces of evolution. How is it that the inherited variation is chosen? In other words, there's some way in which different versions of variation are preferred, and we will want to explore, how that preference is expressed.